In this video, I'll be continuing a review of Chapter 17 of Human Anatomy and Physiology 11th edition. This chapter focuses on the blood. In this video, we'll continue by looking at the erythrocytes and the platelets. We'll look at their structures, functions, and compositions. Let's start with erythrocyte structure. Erythrocytes are typically donut-shaped. This is more technically known as biconcave. You can see that here. It's much easier for me to think of these as donut-shaped. They very much look like donuts with sort of a concave center with a ring around the outside. You'll notice that there is no nucleus within this cell and essentially no organelles. They really have one purpose, and they have multiple, but one major purpose, and that is to carry concentrated hemoglobin around the body. So this red color is due to very concentrated hemoglobin protein within that erythrocyte. And this is the major factor contributing to blood viscosity. So take a look at that word viscosity. That's basically the thickness or the gooiness of blood. And so if you remember, erythrocytes are 45% of your blood volume typically. And so that's going to contribute a lot to how solid your blood is. When you look within there, I'd mentioned that mainly these erythrocytes are hemoglobin carrying machines. And so that hemoglobin is very important to your survival because it is what allows your blood to carry oxygen to tissues. The hemoglobin itself looks like this. It actually has 4 polypeptides that join together. So 1, 2, 3, 4. And these are all joining together to form the full hemoglobin protein. That means each hemoglobin protein can actually carry 4 oxygen molecules. So each of the polypeptides within the hemoglobin can carry one of those oxygen groups. And there are about 250 million hemoglobin molecules in each erythrocyte. That's why the erythrocytes are red, and that's actually why your blood is red in color. During development, some changes occur in hemoglobin based on the needs of the organism. So in a fetus, there are slightly different needs for the blood. So remember that in that case, the oxygen is actually coming from the parent. It's coming from the placenta. That means that it pays for the hemoglobin in that fetus to be high affinity. So basically that fetus wants to collect as much of the hemoglobin from the parent as possible. And that makes it possible for that fetus to get enough oxygen to feed its own tissues. After birth, there's a different form of hemoglobin. So you can see the fetal hemoglobin is called hemoglobin F, and the adult hemoglobin, or really it's the post-birth hemoglobin, is known as hemoglobin A. And so after birth, the hemoglobin A begins to be formed, and the hemoglobin F typically disappears. Hemoglobin A has a lower affinity for oxygen. This actually is very good for your body because it means that that hemoglobin more efficiently releases oxygen at the tissues that need oxygen. So in this case, you're responsible for getting that oxygen from the atmosphere into your lungs, into your blood, and then from your blood to your tissues. And so it's important that the release of oxygen be very efficient. So hemoglobin A is found after birth. There are also some disorders associated with red blood cells. Anemia is an erythrocyte disorder. And anemia just generally describes a condition in which the blood has inadequate oxygen carrying capacity. So since hemoglobin is the major carrier of oxygen in your blood, it means that if there are any issues with hemoglobin or with the red blood cells, this is going to affect your ability to get oxygen to your tissues. There are 3 broad causes of anemia. These can include blood loss. So if you don't have enough red blood cells, that means you don't have enough hemoglobin. Not enough red blood cell production. Again, not enough red blood cells means not enough hemoglobin. You could broaden this out even further to say that maybe there's some issue with the hemoglobin being produced within the erythrocytes you actually have. That could be another cause within that second category. So just not enough red blood cell production or too much red blood cell destruction. There are two major diseases in which this occurs. One is thalassemia, and the other is sickle cell anemia. In both of those, you see excessive destruction. So you're producing plenty of red blood cells, they're just getting destroyed more quickly than they normally would. So rather than living for 100 or 120 days in your body, these red blood cells are being destroyed much sooner. So again, your production isn't enough to keep up with the destruction. Speaking of formation of red blood cells, it has this wonderful word associated with it, which is hematopoiesis. So it's birth of blood cells, or birth of blood, hematopoiesis. And this typically happens in the red bone marrow in adults. So that bone marrow, the red bone marrow in adults, is found in the axial skeleton. It's also found in limb girdles and the proximal epiphyses of the femur and the humerus. So pretty limited in scope where you're going to find it, but this is where red blood cell formation is happening. All blood cells are derived from the same stem cell, and this is called a hematopoietic stem cell. This is going to include hematopoiesis, erythropoiesis, which is the formation of erythrocytes, as well as leukopoiesis, which is the formation of leukocytes. So here we have some pretty hefty vocab words that you might just want to familiarize yourself with. All of them are related to all of them are related to blood cell formation, which is hematopoiesis, and that includes red blood cell formation or erythrocyte formation, erythropoiesis, as well as white blood cell formation or leukocyte formation, which is leukopoiesis. So one thing you could do is just take note that whenever you see this crazy set of vowels with a P and a couple of S's in it, poiesis, that is referring to the formation of something. And in this case, in this unit, it would be the formation of blood cells. So let's look at erythropoiesis a little more closely. So you can see here that we're going through 5 major stages in this developmental pathway. You start with that hematopoietic stem cell. Remember, this is the source for all blood cells, including erythrocytes. In this case, that hematopoietic stem cell is going to differentiate. It's going to differentiate into something called a proerythroblast. So at this point, this cell is now committed to becoming an erythrocyte. But it's still pretty far. You'll notice, for instance, that proerythroblast still has a nucleus in the center of it. So next, it's going to further differentiate into multiple stages of this thing called an erythroblast. So these are just different developmental stages. You can see that the shape of the cell starts to change. The cell itself appears to start shrinking. But as far as this goes, we're seeing this nucleus until the very end of this erythroblast phase. And it's at the end of that erythroblast phase that the nucleus is ejected and that erythroblast becomes a reticulocyte. So now this is the final stage of development, this reticulocyte, before the cell finally develops into a mature erythrocyte. And you can see there it's got that donut shape. It has no nucleus. It's red because of all the hemoglobin forming within it. That's erythropoiesis. So the big note here, 5 steps. You've got that hemopoietic stem cell into the proerythroblast. And then the erythroblast is where you have a lot of that development happening, including and up to the ejection of the nucleus. And so after that ejection of the nucleus, it's the reticulocyte, and then finally mature erythroblast. Here we go with platelets. So platelets, you can see they start again with that hematopoietic stem cell. They're going to go along a different developmental pathway. So instead of Like here with erythropoiesis, instead of developing into this proerythroblast, with platelets, it goes along a different pathway. And this stem cell develops into a megakaryoblast, and then to a megakaryocyte. And so that a megakaryocyte, it's a huge cell, and you can see even in this final stage here, stage 4, it's starting to bleb off pieces of membrane. And that's actually what forms the platelets. It is pieces of this huge megakaryocyte that are fragmenting into platelets. So these platelets are short-lived, so they're going to degenerate within about 10 days after being formed. When you see them, they're typically stained so that they look blue with little purple granules, and they're going to be really important in the blood clotting process. So now I'm going to leave you with a question, and that is, which part of an erythrocyte binds best to oxygen?